what is bioenergetic? As I said, bioenergetic is a study of living organism, that system and or the study of energy in living systems and the organism, for example plants and animals, that utilize them. Okay, this is about the what is bioenergetic, study of energy in living system. Energy required by the all organism may be a kinetic or potential energy. Okay, so this is all about this. Now law of thermodynamics. First law of thermodynamics, also known as law of conservation of energy. What it states, or it is stated that the energy can neither be created nor destroyed. It may change from one form to the another, but the energy in a closed system remain constant. In any physical conditions or physical change, or a total amount of energy in the universe remain constant, although the form of energy may change. Okay, so we will see what are the reactions involved, endergonic and exergonics, and what are the changes found in that. This is the first comparison tables of endergonic and exergonic reactions. For example, energy level of any reactant or any product in endergonic reaction, it is very low, whereas in case of energy level of product, the endergonic reaction have a high. If we see the free energy delta G of the reaction, that is positive in endergonic reaction, whereas in case of exergonic reaction, it is found to be negative. Same thing is the direction in which reaction proceeds spontaneously. Endergonic reaction, that is backward, and exergonic reaction is forward. So that can be asked in the examination as what is the difference between endergonic reaction and exergonic reactions. So you have to write these 4 points with this. Okay. Now we will start with the another comparisons. Same reaction is that first, as I started in the first lecture, what is catabolism, what is anabolism. Same thing is here, but here is the description of the all overall process is that. Okay, and what are the description of the chemical participants involved in that is also given. So first reaction, you have to classify it into this 6-part purpose, nature of the process, energetic, type of starting material, type of final product, typically coenzyme and co-substrate and transform. Okay, this is all about the catabolism and anabolism. Now how it is differ from purposefully, if you see the purpose, energy generation is found in the catabolism, whereas formation of useful compound is found in the anabolism. So the catabolism, energy generation, anabolism, formation of useful compound. Now the nature process or nature of the catabolism process is oxidative or degradative, that I already been taught to you. So anabolism, generally it is the reductive or synthetic, and energetic, that yield energy in the catabolism, and whereas in synthesis or that uses energy. Then type of starting material, that is highly variable of more complex, that is in a catabolism require polymer units, whereas in case of relatively few or simple structure is found in the anabolisms. So types of final product, you will find a polymer into a monomer. Relatively few simple structure, here is monomer into a polymer unit. So highly variable often complex compound is found in the anabolism, whereas typically coenzyme and co-substrate transformation. For example, as I said, energy generation means ADP converted into ATP, and NADP converted into a NADH, and ATP degraded into a ADP, and NADPH reduced into a NADP. Okay, this is all about the anabolism and catabolisms. And in previous slide we have understand endergonic and exergonic reactions. Now we will understand TCA cycles. This is the intermediate pathways you will find. Okay, so understand these. You have to understand this TCA cycle enzymes, catalyzed reaction. In TCA cycles, ACO is Kumeu, okay, which consume or generate a compound of special interest, and these degradations or synthesis you will find. So stages of TCA cycles, there are according to types of reaction catalyzed, that may be a changes of number of carbon in substrate, oxidation of substrate, or production of reduced equivalent, or production of high energy. That is there are 6 various enzymes that involve. Generally 8 enzymes are involved in TAN reaction of TCA cycle. So you have to understand here that first enzyme, citrate synthesis. Here is the addition of C from this acetyl-CoA. Another enzyme, that is dehydrogenase is the enzyme that will found in the oxidative decarboxylation of isocitrate dehydrogenase and alpha-ketobutyrate dehydrogenase. So the removal of carbon is CO2 and oxidation of substrate and production of reducing equivalent, that is NADH. So always you will find NAD converted to NADH or decarboxylations, that is found dehydrogenation enzymes. Now the dehydrogenase, wherever you have NAD found, you have to write in this. Succinyl CoA converts CoA synthetase and succinate dehydrogenase and malate dehydrogenase. Now succinate to fumarate, fumarate to malate, malate to oxaloacetate. Okay, and that citrate, isocitrate, cis-acotinate. Okay, these are the general cycles in which these are the generation of oxidations of substrate, production of reducing equivalent, that is FADH2 in those succinate dehydrogenation. And this is third phase. First is acetate entry, then oxidative decarboxylation, and third is regeneration of oxaloacetate. This is the TCA cycle intermediates and how the energy is generated and what is the bioenergetic behind these cycles. Now the cellular respirations or fermentation inputs or output chart. This is the answer key. You know, generally these are the processes: glycolysis, pyruvate to acetyl-CoA and citric acid cycle. We measure carbohydrate metabolism. Now the location of the glycolysis is cytosol, and pyruvate to acetyl-CoA intermediate step that is found in the mitochondria, and citric acid cycle or great cycle that is in the mitochondrial matrix. So the major functions that begins CR process, catabolic reactions is that, then splitting of sugar is there. So there are 10 reactions found in that glycolysis from glucose to glucose 1,6-diphosphate, glucose 6-phosphate, then fructose 6-phosphate, then fructose 1,6-diphosphate, then that way of phase and preparatory phase. This will gives you a input of 2 ADP and 2 NAD glucose, that will give you overall 2 pyruvate, 2 ATP and 2 NADH. Okay. Similarly, if we see the pyruvate to acetyl-CoA in the mitochondrial matrix, the convert of pyruvate to acetyl-CoA for the citric acid cycle, that input is required is 2 pyruvate, 2 CoA and 2 NAD. Whereas in case of that output, you will get 2 acetyl-CoA, 2 CO2 and 2 NADH. This is the reaction in which what is input and what is output. Now you have to understand what will happen in the citric acid cycle, mitochondrial matrix, that is a complete oxidation of the glucose, that is the energy molecules. This is the major functions of the citric acid cycle. Whereas input is only a 2 acetyl-CoA. Now you see what is the overall output comes in that is 4 CO2, 2 ATP and 6 NAD. This relay electron extracted from the food to electron transport chain to FADH. Okay, now we will continue with this. is the carbohydrate metabolism? If it is that, then you will certainly understand or certainly write only carbohydrate metabolism means you will enlist various pathways, glycolysis, mechanical phosphate pathway, citric acid cycle. No. What is carbohydrate metabolism? Is a fundamental biochemical process that ensures a constant supply of the energy to the living systems. Constant supply of energy. Okay, this is the major functions. Now, metabolic pathways, the process of making and breaking down the sugar molecules, that illustrate two types of metabolic pathway. Metabolic pathway is a series of interconnected biochemical reactions that convert a substrate molecules or molecules step by step through the series of metabolic intermediates. So this is about the carbohydrate metabolism and what are the metabolic pathways and how the reaction is happening. In between I am taking, as I said previously, catabolism and metabolism differences. Now catabolic reaction and anabolic reactions. Okay. So as I said previously, that is the complex and larger molecules are broken down into a simple or smaller during this reaction, that is called a catabolic reaction. In anabolic reaction, you will find the complex molecules that is required for the functionality of body to synthesize and using the simple molecules in this reaction. Whereas in catabolic reaction, this kind of reaction involves the hormone like cortisol, glucagon, cytosine, adrenaline in the human body. This kind of reaction involves hormones like insulin, plant growth hormones, testosterone and estrogen. This difference is catabolic and anabolic reaction of human beings, but you have to understand as a plant. Okay, energy is released during these reactions means catabolic. Energy is utilized during these reactions means anabolic. Okay, that can be asked in the multiple choice. 2. Potential energy is converted into a kinetic energy during catabolic reactions. So that can be asked as a fill in the blanks. So potential energy is converted into a kinetic energy during blanks reactions. So you have to write a catabolic reaction. Same thing, this kinetic energy is converted into a potential energy during blanks reaction, that is anabolic reaction. Now example, you know, one is breakdown, another is synthesis. Catabolic means breakdown and anabolic means synthesis. So protein into a amino acid, that is called catabolic reaction, and synthesis of polypeptide by using a different amino acid into a protein, that is called anabolic reactions. Now we will start with as I repeatedly given a difference between endergonic and exergonic. Now we have to understand what is endergonic reaction. Chemical reaction that require a net input of energy, that is called endergonic. Example, photosynthesis, 6CO2 plus 6H2O, C6H12O, 6 plus CO2 in the presence of light, in the presence of rubisco, that will convert into a carbon dioxide, convert into a glucose molecules and oxygen. Okay, we will start with later on. Thank you.